Hi everyone, let's start the presentation of Group 11. First of all, I'll tell you the most important thing in this video. It is the name. Some of you may think that the title of this presentation is League 1, but it is wrong. The name is League I. Please memorize it. Then, I'll introduce what League I is. The formal name is retinoic acid inducible gene I. You don't have to memorize it, but you can understand that it is protein from its name. It works on innate immunity. Next, I'll tell you about the biological effect. First, it can detect single-strand RNA virus like paramyxovirus. Second, it suppresses the replication of the virus. Finally, it can enhance the killing ability of immune-competent cells like NK cell and TC cell. Then, I'll tell you about the structure of Lig I. Lig I is protein, so it has N-terminus and C-terminus. In N-terminus, it has two CARDs, caspase recruitment domain. In C-terminus, it has one RD, repressor domain. Also, there is RNA helicase domain in the center of Lig I. Usually, Lig I is inactive. CARDs are covered with RD. When Lig I detects virus, it changes to active form with ATP. When Lig I is active, it moves to the mitochondria in the cell and combines with IPS1, which is adaptor molecule. Then, it releases IRF3. It is transcription factor. IRF3 moves to the nucleus and transcribes cytokines, especially interferon alpha and interferon beta. They are called type 1 interferon. They suppress the replication of the virus.
What would happen if Lig I does not exist? Experiments with Lig I knockout mice have shown that if Lig I did not exist, it would not be resistant to single standard RNA virus, such as rabies virus and influenza virus. Lig I is not sensitive to picoronavirus, and these virus are not recognized by Lig I because of the loss of the 5'-terminal triphosphate. However, this is replaced by MDA5. MDA is the same as Lig I. The structure is so similar. So, what does Lig I recognize as the ligand? Lig I recognizes the triphosphate at the 5' end of the viral RNA. Finally, I'd like to explain the relationship between Lig I and SARS-CoV-2 that I've described so far. Surprisingly, our school, Hokkaido University, is conducting the most advanced research on this subject in Japan. When Lig I recognizes the SARS-CoV-2, it will suppress the growth of the virus. But unlike other viruses, it does not work through direct cytokines. This concludes our group presentation. Thank you for your attention.